In this video, I'm going to discuss whether you could restore dinosaur DNA with PCR. Okay, well, the short answer is no, but the long answer is a lot more interesting. I'm going to be breaking this down to 4 parts. What is PCR? How is this relevant to paleontology? How have we used it already? And what can we do to push the limits? PCR is a lab technique that essentially lets us copy and paste DNA. The specific sequence is selected and then broken apart and put back together over and over and over. This is useful in diagnostics, forensics, genetic engineering. Anything with DNA, you will probably see PCR. It has some very strict laboratory conditions which will affect a lot of what we talk about later. Okay, now that I'm done rambling, here's some clear and concise information on what PCR is.
PCR, polymerase chain reaction, is the amplification of specific DNA sequences. It begins with denaturation, where the sample is heated to 95 degrees Celsius, which disrupts its hydrogen bonds. This is followed by annealing, where the sample is cooled from anywhere between 50 to 70 degrees. The final phase is the extension phase. The temperature is raised again between 75 and 80 degrees. DNA polymerase binds nucleotides to primers. This results in the synthesis of a new strand complementary to the template.
I don't know, but I've been told. Land of the world be mighty cold.
Now you're probably wondering what any of this has to do with paleontology, the study of plant and animal fossils. In these fossils is DNA, which is degraded over the millions of years. To make these traces of DNA useful, PCR replicates the DNA, amplifying it to a usable level. With these completed strands of DNA, we get a clearer picture of what our ancestors looked like, as well as what they hunted and ate.
Fossilization begins with a deceased organism. It is buried by sediment and its soft tissue decays, leaving bones and shells. Within the fossil, DNA will rapidly deteriorate, with a half-life averaging 500 years. Its genetic material will eventually be reduced to nothing, leaving morphological analysis as the only insight into the specimen's nature. Once the rocks and landmass burying the fossil have eroded, the specimen can be gathered.
Fossilization isn't the best preserver of DNA, but plenty of prehistoric specimens have been recovered with DNA still intact. From ancient humans to mammoths to giant sloths. Using genetic engineering, there have been attempts to bring back some of these animals, thankfully not the giant sloth. A frightening example is the dire wolf, which was brought back from extinction using genetic modification. This is relevant because they amplified the DNA that was then inserted into the grey wolves. Now for some information on what makes a fossil usable or not.
The conditions for DNA extraction and amplification are dependent on the environment of the fossil and the age of the specimen. Minerals can seep into the structure and contaminate genetic material. However, some can assist in preservation. Neutral or acceptably alkaline conditions are best, while acids lead to erosion through the absorption of microbes. Older fossils have far greater contamination and thus contain many lesions that block DNA polymerase from amplifying and sequencing their strands.
Release the baby! Get ready, go!
Now the question is, what do we do now? We can analyze prehistoric organisms. What do we do with the information we've gathered? What's the point? Even after bringing back dire wolves, most of the DNA had decayed to the point it couldn't be recovered, so they had to fill in the blanks using the grey wolves. What you have technically isn't a dire wolf, it's just a grey wolf with dire wolf characteristics. Of course, you could argue if it walks like a duck, talks like a duck, it's a duck. It looks like a dire wolf, it behaves like a dire wolf, it has the DNA of a dire wolf. At what point does it become a dire wolf? So let's circle all the way back to the beginning. Dinosaurs. There's no DNA left. It's gone through its half-life 100 times over. What if we just make up our own based off the skeletal remains? We create entire sets of complete dinosaur DNA. It's not the same prehistoric creature, but it's identical in every sense. Is that even a dinosaur anymore? In conclusion, while bringing back the dinosaurs is a total long shot, PCR is still extremely useful in paleontology and will be used to replicate older and older and older stuff. A big thank you to Tom C, Sandy Ra, and LSaunders. They did a great job making this sound much more professional. They'll be linked below along with all the references, and thank you for watching.